Hello, my name is Claire O'Haraud, and I'm currently working towards my PhD investigating the potential of biopesticides to control the cabbage stem flea beetle in oilseed rape crops at Harper Adams University. This project is sponsored by AHDB, Scottish Europe, and the Agri-Food Charities Partnership, and in collaboration with the University of Warwick. The aim of my PhD is to identify alternatives to conventional synthetic insecticides, including the use of microorganisms such as fungi, bacteria, as well as nematodes and plant extracts. This project is needed because since the ban by the European Union in 2013 of neonicotinoid insecticides, the control of the cabbage stem flea beetles in oilseed rape crops has been reliant on pyrethroid insecticides. However, populations of cabbage stem flea beetle are now largely resistant to this class of insecticides, and pyrethroids are known to be harmful to naturally occurring predators and parasitoids that may help to reduce cabbage stem flea beetle populations. Alternatives are then needed to control efficiently the cabbage stem flea beetle in oilseed rape crops. My project started in September 2019 and will end in September 2022 after 3 years. So far, the results from my work are that nematodes show potential for controlling cabbage stem flea beetle. I am presenting here the results of a laboratory bioassay that I completed in October with 4 nematode species: Steinonema feltiae, Steinonema capocapsae, Steinonema crossae, and Heterorhabditis bacteriophora at 3 concentrations: 4,000, 10,000, and 40,000 nematodes per milliliter of water on batches of 10 adult cabbage stem flea beetles. Cumulative mortality of the cabbage stem flea beetles was assessed 2, 4, and 6 days after they were treated with the nematodes. We can see that the results are quite variable, particularly when the lower doses of nematodes were used. However, it is clear from these results that the nematode species Heterorhabditis bacteriophora is the most promising species tested, with almost 75% of the cabbage stem flea beetles dead 6 days after treatment at the lowest dose tested, more than 75% dead after only 2 days at the second highest dose, and 100% mortality after only 2 days at the highest dose. The nematode species Steinonema feltiae also performed well at the highest dose, with around 85% of mortality recorded after only 2 days post-treatment. Steinonema capocapsae performed well at lower nematode doses, even at the lowest dose with almost 65% of mortality, but was slower to kill the beetles. Steinonema crossae showed a high variability among replicates and was the least effective of the nematode species tested, killing fewer than 75% of the beetles at any nematode dose. While these results are encouraging, the next step will be to test these nematodes under more realistic crop conditions. I have also completed a laboratory bioassay testing the physically acting products Flipper, developed by Bayer, which is a fatty acids-based product currently used in horticultural crops against whiteflies, aphids, and mites, and another physically acting product coded CEU 40640 ISL and currently being developed by Scottish Europe BV. I tested these products at 3 doses, half the field rate, and double the field rate on batches of 10 adult cabbage stem flea beetles. The cumulative mortality of the beetles was recorded 1, 2, and 5 days after treatment application. Both products showed encouraging results in this laboratory bioassay, with all doses significantly different from water control, for which no death was recorded. Flipper in particular was very effective, with more than 85% of mortality recorded just 1 day after application at the field rate of 10 ml per liter of water, and 100% mortality when the field rate is doubled. It is worth noting that these products act very fast, with no significant differences between cumulative mortality at 1, 2, or 5 days, and maximum effects then reached within 24 hours. As with the nematode results, the next step will be to test these physically acting products under more realistic crop conditions. Over the next few weeks, I am planning to complete other bioassays to test the effectiveness of the bacteria Bacillus thuringiensis tenebrionis, as well as commercial formulations of the fungi Metarhizium anisopliae and Bovia bassiana, and a biopolymer based on linseed oil. You can find out more information by following me on Twitter or visiting the AHDB website. If by any chance you have altered row crops that have been heavily infested by cabbage and flea beetle larvae, please do get in touch. Thank you.